big picture, there's information overload. So your parents' tax dollars have gone to fund lots of research. The NIH budget is billions of dollars. There's a National Science Foundation. There's European equivalents. There's Japanese equivalents. There's lots of money that goes into research. And most of you guys have probably had a class where you had to use PubMed. So the National Library of Medicine has a website where they essentially have a catalog of all these scientific papers that are out there. And there are literally millions of papers that have been published. But if you can't find the information, it's gotten to the point where no human being can know all the information that's out there, that investment is kind of being underutilized, right? So what you'd like to have is ways in which computers can help you with getting that information back out and making it findable and accessible and reusable and so on. Okay, so there's this overwhelming amount of literature that no human can deal with. Computers will be needed to help to work with it, but computers are actually really bad at reading. Okay, it's these days with Alexa and Siri and all those sorts of things, the computers seem like they're getting smarter, but they're actually still really pretty bad at understanding things they haven't seen before and reading scientific papers. So there are professional curators who read the literature and create simpler versions of the information there that the computers can actually understand. And that's part of what you're going to be doing for the function of genes in this project. Okay, so biocurators bridge this gap between human written literature and databases that are read by computers. Okay, so essentially you have a whole bunch of papers or data sets, and you have this rate limiting step, which is these people. This is a conference of biocurators. It's a nice picture that's taken. I'm in there somewhere. And then the curators create records that go into databases, which are much more easily searchable and analyzable by the computers. Okay, so the goal is that you're going to take on the role of being a biocurator. So biocurators actually are, like I said, it's a job, it's a career. There's lots, we're going to share these slides with you guys right away, but there's these different things that biocurators do. There's this International Society for Biocuration, and there's like papers about what biocurators do in terms of their careers. Okay. These are some of the people who employ biocurators. These are like organizations, many of whom you've never heard of. But for example, the Gene Ontology is the consortium of groups that gather information about gene function from all different organisms. We're actually considered, at A&M, we're considered the experts on bacterial gene function. Our function Gene Ontology Consortium is to talk to all these people who work on mice and fruit flies and things like that and say, hey, bacteria do that too. Or bacteria do even more. So, because we're kind of microbiology centered. But like, this is a database that works on cereal plants. This is one that works on budding yeast. This is from C. elegans, Drosophila. That's Arabidopsis, which is a little weedy plant that a lot of people work on. And so on. And these groups hire people to do this curation. So this is just a recent screenshot that Jolene made that shows that here's all these job openings to work at these different databases. Some of them are software engineers, but there's also biocurators. So biocurator, and so on. These are people who are going to be reading information sets, coming up with ways to help them become more computable. Okay, so where are we coming? So this class is based on, the name of the class is something different. I actually never remember the formal name of this class in the catalog, because we all call it the CACAO class. And the reason we call it the CACAO class is it stands for this thing I invented several years ago called the Community Assessment of Community Annotation with Ontologies. So you guys are the community, and you're going to be making annotations with ontologies. And of course, cacao is the plant that chocolate comes from. And so one of the things that this class has a tradition of doing is that at the end of the semester, unless you have some sort of allergy, whoever gets, everybody gets chocolate, and whoever does the best gets to choose first which weird chocolate bars I buy. So there's like, this reward there for us. Okay, so the cacao students then are sort of junior apprentice biocurators. You're going to be doing what some of the professional biocurators do, and with a lot of hand-holding from other people and other instructors. Okay? And the consortium that actually gathers these things professionally, they love us because they figure that you guys are going to be the next generation of scientists who will appreciate the fact that these ontologies exist. You'll know more about how to use them than people who haven't been trained this way. And then just by teaching annotation, it helps us highlight the places where the see, one of the problems with humans is they get to become experts. There's certain shared assumptions they stop thinking about. And so sometimes the people in the consortium have certain assumptions that they share that are not really based on anything. And then when the student says, well, but why is it? Says why, that forces them to re-examine their assumptions, and that's one of the reasons they like us a lot, because we help the consortium re-examine these assumptions. Okay, so your impact. So we're actually in the process of writing a paper about this. Over the years, we've taught this class every semester. Like one of the people who works at the Yeast Genome Database right now at Stanford is somebody who started in your position. He's one of the students in the class who didn't think she'd be able to do it, struggled, and then it took off. She was working in my lab, and she got offered a job at Stanford to work in this database project. And in fact, one of our first-year genetics students is somebody who worked in my lab. And while she was applying to graduate school, we went to a conference, and people at the databases were asking her if she wanted to work with them. So this does have career opportunities as well. Okay, but over the years, we've been teaching this every semester, and students have created thousands of annotations. Right now, there are this many in the database. We actually have submitted more than 5,000. There's a couple of thousand more that are waiting for some guy in Europe to come back from vacation and put them in. We've done this from like 731 different organisms out of like, pushing 3,000 different papers. So students can do this, and you guys will be part of this like line of students who will show that this curation can be done by students. Okay, so what's the impact of this? It makes research more efficient. Okay, it saves a lot of money and resources by helping computers to find the research that people can reuse, and so on. So here's all these different things that are valued by curation. And so the kind of thing that people will do is, what we're going to be doing is we're going to be associating gene products with specific terms that are for function. And it turns out that that's really useful, because then what you can do is if you have a list of gene products that share some common feature, like they're all expressed when you treat with a particular drug, or they're all in a particular tissue type, or they're all in a particular clade in evolution, you can take that list of genes and you can say, what are the functions that are associated with those genes? And there will be some that have known functions that come from annotation. There will be others that are where you don't know anything about their function. And you can guess, well, if these things all have the same biological response, maybe they're involved in the same function. That's called guilt by association, right? This gene is kind of hanging out with these other genes that do this thing. That means maybe it's involved in that same process. So scientists use this a lot. It's just when they do studies of gene expression, evolution, protein complexes, and so on. It's a way of assigning function to unknown genes of unknown function from things that you already know. It's a hypothesis generator. There's so many experiments to test it. It helps you narrow down the possibilities a lot. So this is from a figure, this is from an example where people use these different annotations over there to guess about what the functions were of a particular set of genes. But these actually came from a proteomics experiment looking at extracellular vesicles. We feel like microbes need more representation in Go, because a lot of these different databases that are hiring crowd creators, they're funded to work on eukaryotes, and so we do a lot of microbial stuff. So this is the representation of things that students have done in the past, and I can't pointer, right? So still the majority are eukaryotes, but it's this compared to like in all of the Go consortium, like 95% of what's out there is done for eukaryotes. And so we've increased the amount of annotation for bacteria, and we've done a lot for viruses, which is because Texas A&M is also the home for the Center for Phage Technology. This is a paper reading course. Some of you may have had other paper reading courses, maybe not, or had paper assignments in some course. You didn't have any, you had paper reading and papers in class. Okay. This class, I think, is different than other paper reading classes. Many faculty want to put papers into their classes based on the idea that learning how to read papers is an important skill, even if you're not going to become a professional scientist. But this class is different in several ways. One is that most of the time in the past, in fact, the other class that you're going to take from me, which is based on paper reading. Most of the time it's, I'm the instructor, I picked this paper because I think it's cool, and I want you to read my mind about why I think it's cool. Right? That's what describes the paper reading experience you have in those classes. Right? Write a report, and here's, I'm going to check off whether you saw all the same things I want you to see. Right? This is different because you pick the papers. And what you're going to be graded on is your ability to translate the information in the papers that you picked into this format for CACAO. Okay? It's also different because it has a real world impact. Like I said, I've said several times, we submit the annotations back to the consortium. Most of the classes, if you read the paper, no one else in the world is going to care. That's how that seems. Okay? And it's also, we do it in a real world way where unless you're a really good computer hacker and you can get into our database and change things, you know, in the underlying website, you pretty much can't cheat. Like asking for help is not cheating in the real world, right? It's not like you're locked away in the room with a proctor who says you can't like consult the internet or talk to some human. In the real world, you know, if I don't understand something about, you know, zebrafish, I go to Dr. Riley over in biology and say, hey, I don't understand this thing about zebrafish, can you help me out? And he says, sure, right? Or if I want to learn something about, you know, aspergillus, I go to Dr. Armayo, or if I go to, you know, if I want to learn something about some virus, I go to Dr. Leibowitz in the medical school. So you guys probably know people through the fact that genetics, biochemistry students are all part of your research. So you may have a research advisor, you may have graduate students, postdocs, people down the hall. There's lots of ways to get help that are not cheating in this class, but you might be considered cheating in some other class. So, like I said, it's technically possible to cheat if you're like a hacker and you change the database so the scores all change. But all those other things are not cheating. Okay? The other thing is that there's no partial credit for any annotation you get. So that's how it makes it sound scary. No partial credit. But you can make as many annotations as you want. So if you have to have a perfect annotation in order to get credit for it for class, but if you miss one, you can either fix it or find a different one. Okay, now in addition to your course grade, there's a competition grade, you know, standing, which is what's going to show on the website, which is based on your annotation points. Okay, and that's what determines who gets to pick who takes the instructor.